Okay, these are cell communication notes, cell signaling notes. These notes go into your AP Bio Spiral notebook. In the textbook, reading over 5.6 in Chapter 5 would also be helpful. So cells can communicate either locally or over a long distance. So local communication would be like students sitting at a desk. They physically hand a note from one student to the next because they're nearby and they're physically giving that information. So local communication, one cell secretes a chemical that acts on nearby cells. So for local communication, one cell secretes a chemical that acts on nearby cells. Example, a damaged cell is going to release histamine. That histamine creates inflammation and vasodilation. So an example is a damaged cell releases histamine, histamine. That creates inflammation and vasodilation. Inflammation and vasodilation, vasodilatation. Another example would be an antigen presenting cell. So that cell is able to communicate with nearby cells because it has on its surface an antigen that it wants other cells to recognize. Because it's on the surface of the cell, they're able to get that information easily. But then they do have to be local, it have to be nearby. Another example of local communication, the nervous system releases neurotransmitters. The neurotransmitters are chemicals that move from one neuron to another connected neuron. So another example of local communication, the nervous system releasing neurotransmitters. The neurotransmitters are chemicals that move from one neuron to another connected neuron. So the neurons still have to be nearby, but it's very specific communication. So it's kind of like if you send an email, you're sending that email to a specific email address. So information goes from one cell to another specific cell. Long distance communication. The endocrine system uses hormones to communicate, and this is considered long-distance communication. The endocrine system uses hormones to communicate. So it's not just that the cells aren't nearby, but that's part of it as well. But it's also that your endocrine system is releasing these hormones, and the hormones go all through your body. It goes everywhere in your body. But the hormones are only going to trigger a specific receptor. And so it would be a little bit more like you're posting something to Facebook. That's going to go everywhere. All of your friends are going to see it, but only one specific friend is going to respond to it because you had a comment for that one specific friend. So the hormone goes to all of the cells, and the individual cell decides, sort of, quote unquote, decides if it wants to respond. If it has the appropriate receptor to respond, then it's going to respond to that hormone. We've heard the term ligand before, L-I-G-A-N-D. Ligand is a molecule that bonds specifically to another molecule. A ligand is a molecule that binds specifically to another molecule. Phosphorylation is the addition of a phosphate group. Phosphorylation is the addition of a phosphate group. Phosphorylation. Phosphorylation is the addition of a phosphate group. A protein kinase, ACE tells me it's an enzyme. Whenever I see that word part, I know enzyme. So a protein kinase is an enzyme that uses ATP, which is energy, for this process, for the phosphorylation process. So in order for this to occur, you need a protein kinase, you need an enzyme that can utilize ATP to create the addition of a phosphate group. So protein kinase is an enzyme that uses ATP for the phosphorylation process. All right, so I'll give you the rest of the picture here in just a minute. Phosphorylation cascade. Phosphorylation cascade. A series of chemical reactions during cell signaling mediated by enzymes or kinases, in which each kinase in turn phosphorylates and activates another, ultimately leading to the phosphorylation of many proteins. So cascade, when you think of a cascade, you think of a waterfall. It starts at one spot and then it grows and grows and grows and grows. And that's essentially what's happening here. Phosphorylation cascade, a series of chemical reactions during cell signaling mediated by enzymes, mediated by kinases, in which each kinase in turn phosphorylates and activates another. Ultimately, this leads to the phosphorylation of many proteins. So here's my little picture. This line represents the cell membrane, so outside the cell, inside the cell. I have a receptor that's embedded in my cell membrane. I have a ligand. I have a molecule that is only specifically going to attach to this embedded protein. The ligand attaches to that receptor of that protein, and that causes a signal to be sent inside the cell, and it activates this kinase, which activates those, which activate those, and it causes that signal to multiply within the cell. Signal transduction. A chemical message is converted to actions within a cell. So we're taking a signal, which might be chemical like a ligand, a molecule, could be an electrical signal, but we're changing it into something that can cause an action within the cell. Signal transduction, a chemical message is converted to actions within a cell. So we can modify a protein, we can create a phosphorylation cascade, things like that. So these are all examples of how things go correctly. If we have a faulty pathway, that essentially creates an illness or a disorder. Things aren't working the way they are supposed to be working. So for example, we talked about diabetes, and insulin is the ligand. Insulin is the signal molecule. It's a hormone. It is spread throughout the body until it comes to cells that have the appropriate receptor. Insulin then activates that receptor, and once that receptor is activated, then it opens up a GLUT4 protein, which is basically a door in the cell that lets the sugar in. Think about the things that can happen that would create a faulty pathway and create diabetes. So either the insulin molecule doesn't work, or the receptor doesn't work, or the cell is overstimulated. There's lots of things that can happen, or you're not producing insulin. There's lots of things that can happen that can create that faulty pathway that leads to disease. All right, so the next pages are going to break down those steps. And once again, this is 5.6 in the textbook. All right, so when we talk about local cell signaling, and I've got this divided, not quite into thirds, so, but about the top third, is local cell signaling. Just have to listen to my melodious voice. So, cells are in direct contact. There are gap junctions and plasmodesmata to allow substances to pass freely. So, local cell signaling, cells are in direct contact. Or, for local cell signaling, molecules on the surface of the cell membrane. And that's going to be cell-cell recognition. That's maybe the antigens on the surface of the cell. So we can have cells in direct contact, gap junctions, and plasmodesmata, allowing substances to pass in between those cells. We could have molecules on the surface of the cell membrane for cell-cell recognition. The cells could secrete molecules, so local cell signaling molecules secreted by cells. That would be kind of like that damaged cell releasing the histamine, so molecules secreted by cells. And then neurotransmitters are electrical signals, so local cell signaling, electrical signal between cells trigger release of the neurotransmitters. Electrical signals between cells trigger the release of neurotransmitters. So local cell signaling. Long-distance cell signaling involves hormones in the endocrine system. Long-distance cell signaling involves hormones in the endocrine system. All right, so breaking down the stages of cell signaling. So I've got some color added here because each page after this is going to talk specifically about each stage of cell signaling. So I've got orange for reception information, blue for transduction, green for response information. All right, so these are the 3 steps: reception, transduction, and response. In reception, a molecule binds to a receptor at the cell's surface or inside the cell. So it kind of depends on what kind of ligand it is. Some ligands are only going to be outside of the cell and have to bond to an embedded receptor. Some molecules are small, or they are hydrophobic, so they're able to move in between that phospholipid bilayer, and then they have reception occur inside the cell. So they are ligands that make it inside the cell and bond to their receptor that's inside the cell. Transduction. So this signal that's created when the ligand attaches with the receptor. So this signal is converted to a form that can bring about a specific cellular response. So for transduction, the reception signal is converted to a form that can bring about a specific cellular response. This usually requires a sequence of changes. So signal transduction pathway. Transduction is when this reception signal is converted to a form that can bring about a specific cellular response, and it usually requires a sequence of changes or a signal transduction pathway. Uses relay molecules. So signal transduction pathway, and in parentheses I have here uses relay molecules. So there's relay molecules that are helping that along. And then finally, the last stage is the response. The transduced signal triggers a specific cellular response. So the last stage is response, and here the transduced signal triggers a specific cellular response. So I have a page for reception, a page for transduction, page for response. So let's look at reception first. So this whole page I have reception, and I have a reception on the side there, so I know that this has to do with it. All right. Only a specific signal molecule will bond to and trigger a receptor. Like a hand in glove fit. So there is a specific ligand for a specific receptor. Only a specific signal molecule will bind to and trigger a receptor. It's like the hand in glove fit. The ligand is that signaling molecule. I've seen that term several times, so hopefully we've got that down by now. Okay. Binding of a ligand causes the receptor protein to undergo a shape change in the protein. Binding of the ligand, the signaling molecule, causes the receptor protein to undergo a shape change in the protein. That shape change in the protein can cause receptor activation. The shape change in the protein can cause receptor activation. So I've got a couple of examples here, diagrams, and these are in the textbook. So if you don't like my pictures, you can go to 5.6 and kind of sketch your own. So I have a couple examples of cell surface receptors. So this is the cell membrane right here, this little line. This is my phosphobilipid layer. Here's my cell membrane. Here's before and here's after. So before, I have my ligand. I have my specific receptor. Whenever that ligand bonds to the receptor, it's going to cause a change in the shape of the receptor, which causes that molecule to be released and bond over here. It changes shape, and then it activates this enzyme. So my receptor, once that ligand bonds with it, causes this molecule to be released. This enzyme that is within the cell then changes shape, and it activates that enzyme, and then it can, that's what my little lightning bolts are for. So it's being activated and it can do something. It can now do its job. Another example of a cell surface receptor. So this is an embedded, oh wait, here's my cell membrane, this little line right here. So this is before, this is after. Here's my little cell membrane right here. Can't really see that very well, can you? Okay, cell membrane. And this is an embedded protein, and this is a transport protein. Before the transport protein is closed, it has a spot for the ligand, it has a receptor for the ligand, but the transport protein is closed, so nothing can move in or out. Once that ligand bonds to the receptor, this is that same embedded transport protein, but now it's open. Because it's open, we can have molecules move along their concentration gradient, higher concentration to lower concentration, so it moves into the cell. Ligand-gated ion channel. These are an example of a ligand-gated ion channel. So this ion channel is an embedded protein. Whenever it's closed, things can't move in and out. Whenever it's open, things can move along their concentration gradient. So that ligand, when it bonds to it, it causes it to open. And once it opens, the ions, whatever specific ion that transport protein is specific for, is going to move along its concentration gradient. All right, so these were cell surface receptors. So they're embedded in that plasma membrane, and those ligands don't go into the cell. This other example, this is intracellular receptors. These are receptors that are inside the cell. So these are receptors in the cytoplasm or nucleus of the target cells. Intracellular receptors, that means it's inside the cells. The receptors, they're not on the cell surface like these others. The receptors are in the cytoplasm or the nucleus of the target cells. So inside the cell, in the cytoplasm, or in the nucleus of the cell. So the ligands or molecules are small and or they're hydrophobic, so they can pass through that cell membrane. The ligands / molecules are small and or hydrophobic, so they can pass through the cell membrane. They can turn on genes to be transcribed and made into proteins. So remember, transcription, scribe, you're writing something, so messenger RNA, message gets written. So whenever this happens, it can go in and turn on a gene and tell it to start being transcribed, tell that messenger RNA to be made, which will eventually make it into proteins. So here's my diagram for that. This is the cell. This represents the cell membrane. But this time that ligand is able to move through the cell membrane into the cell. We have a receptor. Receptor in this case is going to be inside the nucleus right here. So that ligand is able to bond to the receptor. Once it bonds to the receptor, it's going to tell that gene and DNA to create messenger RNA. The messenger RNA goes outside of the nucleus into the cytoplasm to where the ribosome is at and then makes protein. Complicated. All right, so that was all reception. Next,